We are a group of biology students studying at Georgia Tech, and we decided to work together with the Piedmont Park Conservancy to study the impact of the runoff from the small dog park on the newly discovered Six Springs wetland.
As you can see, there's a gradual slope from the dog park to the stream. This stream connects the wetlands. The newly built dog park has brought many different dogs to Piedmont Park. The dog park provides a place for dogs to run around and play. However, this can pose some danger for the wetland, as the fecal coliform levels may rise due to dog feces.
E. coli is a type of bacteria found in intestines and feces of animals. It is known as an indicator species, which means that there is a direct correlation between its presence and the presence of pathogens. It's also bad for the biodiversity of soil, showing an inverse success relationship between the bacteria in the soil and E. coli. While most strains are innocent, contamination through direct contact can make you sick. Symptoms are similar to those of food poisoning.
So this is an image of the small dog park, and basically our hypothesis was that as the dog park is going downhill due to runoff, E. coli will be mostly deposited here where it levels out. Basically for our methods, we took 8 samples in 4 different locations, here, 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 and here, and we also took a set of control samples above the dog park as well.
To start our experiment, we selected our sample sites, choosing to take samples from 8 sites across 4 levels. We then took our samples, placing them into metal tins, and took our samples back to the lab, where we measured out 10 grams of each sample and diluted our soil and water. We then placed a filter on top of a vacuum and used vacuum filtration to filter out bacteria from our samples. The filter was too small for the bacteria to pass through, but easily allowed water to pass through, allowing us to catch the bacteria on the filter. We then took the filter and placed it in a petri dish full of Hock M. coli blue broth, which color-coded the bacteria while suppressing other bacteria from growing. We incubated the petri dishes at 35 degrees Celsius for 24 hours and then counted the colonies present. This is what the colonies looked like after incubation. As you can see, some colonies are blue and some are red. The red colonies indicate E. coli specifically, while the blue colonies indicate fecal coliforms.
After plating our soil samples, we found that the colony count increased as we went down the slope of the dog park. Found particularly high concentrations here, as we expected, but we also found high concentrations across the stream, and we think this is because there are other animals that live there. There is also runoff from different areas of the park that are depositing here as well.
This diagram shows the number of colonies found in the areas around the dog park. Darker colors indicate higher numbers of colonies, while lighter colors indicate lower numbers of colonies. The higher colonies were found in the drainage area of the dog park in the area just past the fence. This graph indicates the same, showing that the highest levels were found before the wetland. Our results indicate that there is contamination of the wetland from the runoff from the dog park. The number one thing that you can do to help with this is to pick up after your dog and to wash your hands after you leave the park. This will help us keep Piedmont Park clean and safe for everyone.